1.8 billion years ago, Earth had one continent, one ocean, one sky, and almost nothing alive on it had eyes. There was no grass, no trees, no insects, no fish, no soil, just bare rock stretching across a world that would look almost unrecognizable to us. Even the sky was wrong. Scientists still debate its exact color, but they agree on one thing: it was not blue. The atmosphere above this world was a fundamentally different chemical mixture from the one you are breathing right now. The continent was called Columbia. It has also been called Nuna. It was Earth's first confirmed supercontinent, assembled more than a billion years before Pangea and lasting longer than the entire history of animals on land. The world it ruled was equally strange. A day lasted only 18 hours. The moon hung closer and larger in the sky, driving tides far more powerful than those of today. Oxygen levels were only a fraction of modern levels. Ultraviolet radiation struck the surface with an intensity that would be lethal to many modern organisms. There was no soil anywhere on Earth because the life capable of making soil had not yet evolved. And yet life was here, not in a few isolated places, everywhere, invisible to the naked eye, but already reshaping the chemistry of the planet in ways that would eventually make every animal, every forest, and every human life possible. Columbia is not a theory. Geologists have reconstructed it from matching mountain belts, rock formations, and ancient continental fragments now scattered across 6 continents. Most people have heard of Pangaea. Almost nobody has heard of Columbia, which is strange because Columbia helped create the world that made Pangaea possible. Tonight, we're exploring what life was really like on Earth's first supercontinent and the extraordinary world it was quietly building long before anything with eyes existed to see it. Imagine compressing the entire history of Earth into a single long journey and then stepping into it halfway through. That is where Columbia appears. Our planet formed around 4.54 billion years ago. By the time Columbia emerged, roughly 1.9 to 1.8 billion years ago, Earth had already spent more than half of its existence changing shape, cooling, colliding, and rebuilding itself. Countless chapters had already passed before this supercontinent entered the story. The world of Columbia belonged to the Paleoproterozoic, a span of deep time that rarely appears outside geology textbooks. It sat in a distant interval between the stabilization of large continental masses and the arrival of organisms with recognizable bodies. No forests covered the land. No animals crossed shorelines. Even the most familiar forms of life from later ages remained far beyond the horizon. The Earth of this era was governed by processes measured in millions of years, with continents drifting at the pace of growing fingernails and oceans reshaping themselves across immense stretches of time. Before Columbia, the planet's crust was arranged very differently. Large fragments of ancient continental rock, known as cratons, existed as separate worlds. These durable foundations drifted across the mantle beneath them, approaching one another, colliding, and occasionally moving apart again. Each carried its own geological history, preserved in rocks that had survived earlier cycles of planetary change. Although some connections formed temporarily, no single landmass dominated the globe. Columbia marked a turning point in that pattern. For the first time, many of these ancient cratons became joined into a single coherent continental assembly. Geologists classify it as Earth's earliest widely recognized supercontinent. At its greatest extent, it covered roughly 30% of the land surface that exists on Earth today. The exact shape remains the subject of ongoing research, yet the broader picture is clear. Pieces of crust that had spent vast spans of time apart became linked into one immense geological structure. The name itself arrived much later. In 2002, geologist John Rogers and M. Santosh proposed the term Columbia. Part of the inspiration came from the Columbia River region of North America. Another connection came from rocks of the same age found beneath Washington, District of Columbia. Many researchers also use the name Nuna, derived from an Inuit word associated with lands. Both names refer to the same supercontinent, a place known only through fragments preserved in stone. For orientation, Columbia stands at the beginning of a sequence that shaped the long architecture of Earth's continents. After Columbia came Rodinia. After Rodinia came Pannonia. After Pannonia came Pangaea, the supercontinent most people recognize today. Columbia lacks the fame of its younger successors, yet it occupies a special place in the timeline. It was the first known attempt at bringing much of Earth's continental crust together. Its existence stretched across approximately 300 to 500 million years before the bonds holding it together began to weaken. During that immense interval, the planet continued to transform in ways that would echo across deep time. Yet Columbia's story truly begins before its completion, in an age of collisions, pressure, and planetary upheaval that forged the foundations on which the supercontinent would stand. Far beneath the landscapes we know today, there are scars that record one of the greatest construction projects in planetary history. They were carved when continents moved toward one another across disappearing oceans, compressing the crust with forces powerful enough to reshape entire regions of the Earth. Columbia emerged from that prolonged turbulence, assembled through collisions so immense that their traces remain visible nearly 1.8 billion years later. Geologists describe these episodes as orogenic events, periods when converging pieces of crust crumpled, thickened, and rose upward into mountain systems. During the formation of Columbia, such events unfolded across multiple parts of the world within a broadly similar span of geological time. Separate continental fragments approached one another from different directions, each collision contributing another piece to the growing supercontinent. What appears today as a collection of distant continents preserves evidence of a shared chapter written deep within the Paleoproterozoic. Among the most significant of these events was the Trans-Hudson orogeny, which occurred around 1.8 billion years ago. This collision welded together ancient continental cores that would eventually become parts of North America, Greenland, and Europe. Vast ocean basins vanished as crustal blocks converged. Layers of rock folded, fractured, and sank into the depths. The process unfolded over millions of years, producing a durable continental framework that survived long after the mountains themselves disappeared. Comparable transformations were occurring elsewhere. The Svekofenian orogeny shaped portions of what is now Scandinavia. The Capricorn orogeny helped join regions associated with Western Australia and India. Across South America, the Trans-Amazonian orogeny left its own record of compression and crustal growth. Another major collision along the Condylite Belt contributed to the union of the North China Craton with India. Though separated by immense distances, these regions participated in a broader planetary reorganization that gradually stitched Columbia together. The mountain ranges created by these collisions may have rivaled and perhaps exceeded the scale of the modern Himalayas. Crust compressed from opposing directions thickened dramatically, lifting immense belts of rock toward the sky. Conditions differed from those of the present world. Portions of Earth's interior retained greater heat, influencing how the crust responded to pressure. Entire mountain systems rose where oceans had once existed, forming elevated terrain that stretched across continental margins. Those peaks have long since vanished. Nearly 1.8 billion years of weathering and erosion reduced them to their deepest foundations. What remains are the roots. The Canadian Shield preserves fragments of those buried structures. The eastern Ghats of India contain rocks altered during these collisions. Western Australia's Pilbara region holds additional clues. In each case, geologists read the remnants much like pages from an incomplete manuscript. Heat played an equally important role. Rocks trapped deep within thickened crust experienced intense pressure and temperature. Existing minerals became unstable and transformed into new assemblages, creating metamorphic complexes that now serve as geological fingerprints of Columbia's assembly. At the same time, oceanic plates descended beneath continental margins, feeding chains of volcanoes comparable in character to the modern Pacific Ring of Fire. Deep below those volcanic arcs, portions of the crust partially melted, generating large volumes of granite. That granite later cooled and hardened, becoming part of the continental foundations exposed today across shield regions on multiple continents. By the end of this long interval, a supercontinent stood assembled from former fragments, its foundations forged through collision, heat, volcanism, and stone. Above Columbia, daylight would have carried a color no human memory can hold. The land below may have been empty stone, but the first shock of that world would have arrived from overhead, where the atmosphere filtered sunlight through a chemistry unlike the air of the present day. A familiar blue sky would have been unlikely, because the gases and particles above the supercontinent scattered light in a different way. Oxygen existed, though only as a fraction of modern abundance, perhaps around 1 to 4% of present levels. That thin share changed everything. In the oceans and their sediments, methane generating archaea released methane in enormous quantities, feeding the air with a gas that responded strongly to sunlight. Higher in the atmosphere, ultraviolet radiation broke and rearranged methane molecules, helping build complex organic haze particles. These particles gathered into a reddish brown veil, a photochemical smog spread through the upper air. Planetary scientists often compare this kind of sky to Titan, the largest moon of Saturn. Titan is wrapped today in an orange methane haze that turns sunlight into an amber glow before it reaches the surface. Columbia's atmosphere would not have been identical, yet the comparison gives the closest modern doorway into imagining it. The sky may have looked coppery, smoky, or muted by organic aerosols, with the sun shining through a softened, tinted ceiling. The sun itself was different. Around 1.9 to 1.8 billion years ago, it produced only about 85 to 88% of its present luminosity. Stellar aging explains this slow change. Young stars, like the early sun, burn with lower output, then brighten across immense stretches of time as their interiors evolve. From Columbia's surface, sunlight would have been weaker than today, filtered again by haze before touching rock and water. Even with that dimmer sun, Earth did not remain locked in permanent ice. Methane and carbon dioxide helped trap heat, strengthening the greenhouse effect and keeping the planet habitable for microbial life. Some reconstructions suggest global conditions may have been warm, perhaps warmer on average than the present world, though the exact climate remains uncertain. The air above Columbia held a balance between faint sunlight and heat-trapping gases, creating a sky that looked alien while still allowing oceans to persist. Ozone offered little protection. A strong ozone layer requires abundant oxygen, and Columbia's atmosphere had far less oxygen than the air now surrounding Earth. With only a weak shield, the more damaging parts of ultraviolet radiation could reach the surface at intensities hostile to most modern organisms. Deoxyribonucleic acid strands in many surface dwelling life forms would be damaged within minutes under such exposure. Life survived best where water, sediment, or mineral surfaces provided cover. Winds still crossed the supercontinent. Temperature differences between land and surrounding ocean drove moving air across bare plains, volcanic slopes, shorelines, and eroded mountain roots. Those winds carried dust, salt, and the scentless chemistry of a world without forests, flowers, soil, animals, or voices. Above it all, the copper colored sky spread from horizon to horizon, while beyond the coast waited an ocean just as strange. At Columbia's edges, the world opened into one continuous ocean. There was no Atlantic Ocean, no Pacific Ocean, and no Indian Ocean divided into familiar basins. Water wrapped around the supercontinent and entered it through low seaways, flooded margins, and long coastal inlets. From orbit, if any eye had existed to see it, Earth would have shown one great body of water pressed against a single enormous landmass. That ocean would have looked unlike any sea on the planet today. Near the surface, some waters held oxygen released by microbial communities living where sunlight could reach. In those upper layers, aerobic microbes could survive, using oxygen in ways that would later become central to complex life. The chemistry changed with depth. A boundary called the chemocline separated the partly oxygenated surface from water below, where oxygen vanished and dissolved iron remained abundant. For billions of years, iron had entered the ocean from seafloor rocks, volcanic systems, and weathered crust. With limited oxygen available to remove it, the deep sea could store iron in solution. Where that iron rose toward oxygenated zones, chemical reactions transformed it into rust-colored particles. In places near microbial blooms, seawater may have shifted from greenish tones to reddish-brown stains, spreading color through currents with no modern equivalent. The ocean was a chemical landscape as much as a body of water. Some basins carried another harsh signature. Sulfur-reducing bacteria produced hydrogen sulfide, enriching certain waters with a compound poisonous to most modern animals. These sulfidic zones would have been difficult places for familiar life, yet they belonged naturally to Columbia's world. The ocean did not follow the expectations of later seas filled with fish, reefs, and swimming predators. Its structure was layered, reactive, and ruled by microbes able to use chemistry as a source of energy. On the deep sea floor, hydrothermal vents released superheated mineral-rich water into dark basins. Around them lived chemolithotrophs, organisms that gained energy from inorganic chemical reactions rather than sunlight. They drew life from minerals, metals, sulfur compounds, and heat rising through fractured crust. In those places, biology was tied directly to geology. The planet fed its smallest inhabitants through openings in stone. Surface waters in some tropical regions may have reached 40 to 50 degrees Celsius, or 104 to 122 degrees Fahrenheit, according to certain climate models. Such warmth would feel more like a hot bath than a modern sea. Most present-day fish could not survive there, though fish did not yet exist. The life of Columbia's ocean belonged to microbial worlds adapted to heat, chemistry, and changing oxygen levels. The moon also shaped the water. It orbited closer to Earth than it does today, giving its gravity greater influence. Tides may have carried roughly twice the force of modern tides, pushing water across low coastal rock, carving shorelines, and flooding wide tidal flats before withdrawing again. These margins shifted between exposure and submersion, leaving minerals, microbial films, and wet stone beneath the tinted sky. Along a Columbia shore, waves struck billion-year-old rock with nothing living on land to soften their arrival. No forest roots held the coast. No animals moved through the spray. Water, iron, heat, and stone made the rhythm of the planet. Life on Columbia would not have announced itself with movement across the land or shapes drifting through open water. It lived in films, mats, grains, sediments, and suspended cells. Every organism on Earth was microscopic. Every living thing belonged to a scale too small for the naked eye. Yet together they formed an empire spread across shorelines, shallow seas, mineral surfaces, and the upper layers of the ocean. The three great domains of life were already present. Bacteria had diversified into many forms, filling chemical roles across the planet. Archaea, equally old and deeply different in their inner chemistry, occupied environments that would challenge most modern organisms. The earliest members of eukaryota also existed, carrying cellular features that would one day become central to plants, fungi, and animals. At this stage, though, even those distant relatives of complex life remained single-celled and small. Bacterial communities shaped much of Columbia's surface world. Cyanobacteria harvested sunlight in shallow waters, releasing oxygen into their immediate surroundings. Iron oxidizing bacteria lived near chemical boundaries where dissolved iron met oxygen-rich pockets. Sulfur-reducing bacteria worked through sediments where oxygen was absent. Methane-producing archaea occupied deeper layers, helping supply the atmosphere with methane and supporting other microbes that consumed it. Each group held a place in a world organized by chemistry. Archaea thrived in settings that seemed severe by familiar standards. Some lived in hot springs, fed by volcanic heat. Others endured hypersaline lagoons, acidic volcanic pools, and anoxic sediments sealed away from oxygen. Their membranes and enzymes differed from those of bacteria, giving them tools for survival in conditions where ordinary cellular machinery would fail. Columbia's most extreme environments were not empty margins. They were habitats. The total biomass of this world was substantial. Life was not a faint dusting across a hostile planet. Microbial mats spread over wet rock, tidal flats, and shallow seafloor wherever moisture, minerals, and energy allowed them to grow. A square meter of coastal surface could hold dense layered communities, with different microbes arranged by light, oxygen, sulfur, iron, and organic matter. To the eye, such a surface might appear as stain, crust, slime, or color. Biologically, it was a crowded living system. Viruses were also part of this world. Their lineages reached back far earlier than Columbia, and their activity shaped microbial evolution every day. Viral infection moved genetic material between organisms, carrying fragments of deoxyribonucleic acid across species boundaries. Through this horizontal gene transfer, microbes gained new metabolic tools without waiting for ordinary reproduction alone. The invisible empire was constantly editing itself. Its ecology did not resemble an animal food chain. Energy moved through chemical exchanges. Oxygen released by cyanobacteria supported aerobic bacteria, while harming organisms adapted to oxygen-free conditions. Methane from archaea fed methane-consuming microbes. Sulfur compounds passed through several metabolic pathways at once. Iron changed form as microbes and chemistry acted together. This complexity made Columbia's living world chemically rich beyond its visible simplicity. Without animals grazing, burrowing, swimming, or filtering the seas, microbial communities controlled the major exchanges between ocean, atmosphere, and rock. From thin films on stone, they governed the planet's breath. A colony of cells can become a monument if time is allowed to do the building. Along Colombia's shallow coasts, cyanobacteria spread across sunlit rock and sediment in layered mats, each generation adding a little more structure to the last. They were the dominant photosynthetic organisms of this world, and through their ordinary metabolism, they released oxygen into seas that still carried the chemistry of an older Earth. To many organisms that had ruled before them, oxygen was dangerous. It reacted easily, damaging delicate cellular machinery in life adapted to oxygen poor conditions. For cyanobacteria, it was waste from photosynthesis, released after sunlight helped them split water and capture energy. That waste moved outward through the water, changing local chemistry around the mats before spreading farther into coastal seas. The transformation was not dramatic from moment to moment. It came through repetition, cell after cell, day after day, across spans too long for any single shoreline to remember. Their most visible creations were stromatolites. These layered formations grew when microbial mats trapped grains of sediment, bound them with sticky surfaces, and then grew upward over the captured layer. New sediment arrived. New growth sealed it in place. Through thousands to millions of years, the process built domes, columns, and rounded ridges rising from shallow water. Some measured only a few centimeters. Others could reach several meters, becoming biological architecture large enough to shape the look of entire coastal fields. Colombia's shorelines would have held broad stromatolite provinces where sunlight reached the seafloor. Seen from above the water, such places might have appeared as dark, rounded mounds beneath moving ripples clustered across tidal flats and warm lagoons. Tiny oxygen bubbles rose from microbial surfaces during active photosynthesis, slipping upward through water already stained by minerals and chemical gradients. These were living structures, rooted in place, growing by fractions and layers. Stromatolites were chemically powerful. Their oxygen output entered the ocean one molecule at a time, reacting with dissolved iron, altering sediments, and shaping the air above the sea. The change moved across hundreds of millions of years, yet its source could be as thin as a film of cells on stone. Geological events lifted mountains and joined continents, but these microbial builders worked through chemistry, persistence, and light. Their history stretched far deeper than Colombia itself. Fossil stromatolites reach back around 3.5 billion years, making them roughly 1.7 billion years older than this supercontinent. By the time Colombia existed, stromatolites were already survivors from earlier planetary ages. They had endured changing oceans, volcanic eras, shifting atmospheres, and repeated rearrangements of crust. Some still live today, in Shark Bay, Western Australia, and in parts of the Bahamas. Modern stromatolites continue to grow, trap sediment, and photosynthesize. Their structure echoes the older forms that lined Colombia's coasts. They did not need speed, movement, or complexity to succeed. Their method remained effective across nearly unimaginable time. Their dominance would later fade around 1 billion years ago, as early eukaryotic grazers likely began feeding on microbial mats. The architects of Colombia's shores were eventually reduced by the complex life their oxygen helped make possible. Yet in their long prime, they remade the ocean, prepared the atmosphere, and left their layered signature in stone. A strange snowfall drifted through Colombia's seas. No flakes formed in the sky, and no clouds released it. This snowfall began within the water itself, born from a meeting between oxygen and iron that had never occurred on such a scale before. Across broad stretches of the ocean, chemistry was writing one of the most important geological records in Earth's history. The deep waters surrounding Colombia contained enormous quantities of dissolved iron. For billions of years, iron had entered the ocean from hydrothermal vents, volcanic crust, and weathering processes along the sea floor. In a world where oxygen remained scarce, that iron stayed dissolved instead of forming solid minerals. Ocean currents carried it through basin after basin, building a reservoir that had accumulated over immense spans of time. The water itself became a storehouse of metal. Near the surface, cyanobacteria continued releasing oxygen through photosynthesis. Most of that oxygen never reached the atmosphere immediately. Instead, much of it entered the ocean and began reacting with substances already present in the water. As oxygen diffused downward into iron-rich layers, a powerful chemical transformation occurred. Dissolved iron combined with oxygen and formed iron oxide. Rust was appearing throughout the ocean, not on exposed metal surfaces, but suspended within seawater itself. Once formed, these tiny particles could no longer remain dissolved. They drifted downward through the water column in a continuous rain of mineral grains. Day after day, year after year, iron oxide settled onto the seafloor. The process never arrived as a single dramatic event. It unfolded steadily, creating thin deposits that accumulated across vast underwater landscapes. The cycle repeated many times. Oxygen entered a region of water and consumed available dissolved iron. Eventually, the oxygen was exhausted, allowing iron concentrations to build again. A later pulse of oxygen triggered another episode of oxidation. Between the iron-rich deposits, silica settled from seawater as separate layers. Through countless repetitions, the ocean floor accumulated alternating bands of iron-rich rock and silica-rich rock. These deposits became the banded iron formations that geologists study today. Some of these formations reached extraordinary dimensions. In Western Australia's Hamersley Basin, banded iron formations grew to thicknesses approaching 2.5 kilometers, which is about 1.6 miles. Similar deposits formed in the Anamiki Basin around Lake Superior within the Transvaal supergroup of South Africa, the Karajas Formation of Brazil, and the Krivoy Rog Formation of Ukraine. Although separated across today's continents, they preserve evidence of the same global process. These rocks are more than geological curiosities. They represent the physical record of microbial oxygen production operating across hundreds of millions of years. Layer after layer preserves the interaction between life and ocean chemistry. The bands can be read almost like pages in a planetary archive, documenting a transition that reshaped Earth's environment. Their significance extends directly into the modern world. Iron extracted from these formations becomes steel used in buildings, bridges, ships, railways, industrial machinery, and surgical instruments. Material deposited by microscopic organisms nearly 2 billion years ago remains embedded in daily human life. The connection runs from cyanobacterial mats to skyscrapers through an unbroken chain of geological history. Around 1.8 billion years ago, major banded iron formation deposition largely came to an end. Oxygen levels had risen enough to keep much of the ocean's iron continuously oxidized. The chemistry that created those alternating layers could no longer operate in the same way. The rusting ocean had changed, and its striped record remained behind in stone. In a shallow sea somewhere along Colombia's immense coastline, a transformation occurred that left no monument, no crater, and no mountain range behind it. It happened within a single microscopic cell. The event passed unnoticed by the world around it, yet its consequences would eventually reach every forest, every coral reef, every bird, every mammal, and every human being who would ever live. At some point between roughly 2.1 and 1.8 billion years ago, the first true eukaryotic cells emerged. These were cells containing a nucleus, an internal compartment that enclosed and organized genetic material. The appearance of the nucleus marks one of the most significant turning points in biological history. Since the origin of life itself, few innovations have altered the future of the planet more profoundly. Before this transition, all known life was prokaryotic. Bacteria and archaea carried their genetic information without a nucleus, leaving it distributed within the cell's interior. These organisms were highly successful and astonishingly adaptable, yet their cellular architecture imposed limits. Genetic information could be stored, copied, and expressed efficiently, but the level of organization required for much larger and more complex cellular systems had not yet appeared. The nucleus changed that arrangement. A membrane enclosed the genetic material, separating it from the rest of the cell's internal activity. This provided new opportunities for regulating genes, controlling cellular processes, and protecting genetic information. Chromosomes could become larger and more sophisticated. Cellular functions could be coordinated with greater precision. The foundation for biological complexity had been established. Evidence of these early eukaryotes appears in the fossil record during Columbia's era. One of the oldest widely accepted examples is Grypania spiralis, a spiral-shaped organism discovered in rocks from what is now Michigan and dated to approximately 1.87 billion years ago. Although scientists continue to debate aspects of its biology, Grypania is generally regarded as one of the earliest known representatives of eukaryotic life. Its presence shows that the transition had already occurred by that time. The significance of this innovation is difficult to overstate. A typical prokaryotic cell may possess a few thousand genes. Eukaryotic cells can manage many more, organizing them across multiple chromosomes and regulating them through intricate cellular machinery. Such complexity eventually allowed the development of specialized cell types, each performing different roles while remaining part of a larger organism. The distant possibility of muscles, leaves, eyes, and brains begins here. The nucleus itself did not appear suddenly. It emerged through a long sequence of evolutionary refinements involving membranes, genetic organization, and increasingly compartmentalized cellular structures. Hundreds of millions of years of gradual innovation likely preceded the appearance of fully developed eukaryotic cells. By the time they entered the fossil record, much of the underlying work had already been completed. Columbia's oceans, therefore, hosted more than microbial communities, shaping the chemistry of the planet. They also contained the first members of a lineage that would eventually produce every complex organism known. The appearance of the nucleus started a new biological chapter. From that moment onward, evolution possessed a cellular framework capable of supporting levels of complexity that had never before existed on Earth. Every ecosystem on Earth depends on countless acts of consumption. One organism absorbs another. Nutrients are dismantled, recycled, and incorporated into new life. Across billions of years, this pattern remained one of biology's most reliable rules. Cells engulfed other cells, and the engulfed cell disappeared. Somewhere during the interval between roughly 2 billion and 1.45 billion years ago, that familiar outcome failed to occur. A primitive eukaryotic host cell encountered a bacterium belonging to a group known today as alpha proteobacteria. The host engulfed it, drawing it inside its cellular boundary. Under ordinary circumstances, digestion would have followed. Enzymes would have broken the captured organism apart and converted its components into raw materials. Instead, something extraordinary happened. The bacterium survived. More than survival followed. The bacterium continued functioning within its host, carrying out aerobic respiration using oxygen available in the surrounding environment. The host gained access to a powerful new source of energy. The bacterium gained protection and a stable supply of nutrients. Generation after generation passed, and the partnership endured. Eventually, the two organisms became inseparable. This process is known as endosymbiosis, the permanent incorporation of one organism into another living organism. The captured bacterium gradually evolved into the mitochondrion, the energy producing structure present within nearly every eukaryotic cell alive today. What began as a partnership became an inheritance passed from one generation to the next. The significance of mitochondria lies in their efficiency. Through aerobic respiration, they use oxygen to extract energy from nutrients far more effectively than older anaerobic pathways. The molecule produced through this process is adenosine triphosphate, the universal energy currency of cellular life. Oxidative phosphorylation, the mechanism employed by mitochondria, yields roughly 18 times more usable energy than fermentation alone. That increase transformed the possibilities available to life. Large cells became practical, greater genetic complexity became manageable, specialized structures could evolve without exhausting available energy reserves. Every demanding biological activity rests upon this foundation. Muscles contracting, neurons transmitting signals, hearts pumping blood, and lungs expanding all require enormous amounts of cellular energy. Without mitochondria, the energy budget necessary for complex multicellular organisms would not exist. Evidence of this ancient merger remains visible within modern cells. Mitochondria possess their own deoxyribonucleic acid, separate from the deoxyribonucleic acid housed within the nucleus. That genetic material is circular, matching the pattern found in bacteria. Mitochondria reproduce through binary fission, dividing in a manner characteristic of bacterial ancestors. Their biology still carries unmistakable traces of their former independence. A second, endosymbiotic event later reshaped another branch of life. A eukaryotic cell engulfed a cyanobacterium and retained it. This partnership eventually produced chloroplasts, the photosynthetic structures found in plants and algae. Fossil and molecular evidence places this transformation around 1.5 billion years ago. The same broad world that nurtured early eukaryotes also witnessed the emergence of cells capable of harvesting sunlight through a permanently incorporated microbial partner. These events changed the trajectory of life more profoundly than any predator, extinction, or climate shift. The nucleus had organized the cell. Endosymbiosis supplied unprecedented energy and, later, a new way to capture light. Through cooperation preserved across deep time, separate organisms became components of a larger whole, establishing the cellular architecture upon which all complex life would ultimately be built. Imagine measuring time by a sky that moved faster than instinct expected. On Columbia, a full rotation of Earth lasted only about 18 to 19 hours. Dawn arrived sooner, midday came earlier, and evening returned before a modern calendar day would have reached its end. The rhythm of light and darkness belonged to a planet spinning more rapidly than the one we know today. Morning began with a sun that looked subtly unfamiliar. It rose through layers of methane haze and organic particles suspended high in the atmosphere, spreading amber and copper tones across the horizon. The solar disk appeared slightly smaller than the modern sun because the star itself was still earlier in its long life. Its brightness remained substantial, enough to illuminate the landscape clearly. Yet the quality of that light carried a different character. Colors appeared filtered, softened, and tinted by atmospheric chemistry that no longer exists. Higher overhead hung another striking feature. The moon orbited much closer to Earth than it does now, perhaps between 15,000 kilometers, which is about 9,300 miles, and 20,000 kilometers, which is about 12,400 miles, nearer than its present distance. To an observer standing on Colombia's shoreline, it would have appeared noticeably larger in the sky. Its gravitational influence reached deeper into the oceans, pulling water inland with greater force and shaping coastal regions through powerful, recurring tides. Beneath those skies lay terrain untouched by forests, grasses, roots, or burrowing animals. Every step landed directly on exposed stone. There was no true soil, because soil depends on generations of biological activity to break, mix, and enrich mineral material. Sand existed only in limited forms, lacking many of the processes that later organisms would contribute to its production. Rock dominated everything. Some surfaces carried red stains from oxidized iron. Others appeared charcoal black, pale gray, or mottled with mineral deposits left by water and weather. Along the coast, the air likely carried scents unfamiliar to any modern traveler. Marine microbes produced sulfur compounds that drifted inland with sea spray and tidal moisture. Without seaweed, flowering plants, forests, or animal life, none of the familiar coastal fragrances existed. The atmosphere probably possessed a sharper mineral quality, mixed with chemical traces released by microbial ecosystems and volcanic activity. Temperature conditions varied dramatically across the supercontinent. Coastal regions remained moderated by the surrounding ocean and were often warm. Far inland, thousands of kilometers, which is about 1,864 miles, from maritime influence, climates could become extremely dry. Daytime heating and nighttime cooling operated without vegetation to soften their effects. Bare rock absorbed sunlight efficiently and released stored heat after sunset, creating pronounced daily fluctuations. Sound traveled across this landscape in a sparse but powerful way. Wind swept over stone ledges and across open flats. Waves struck rocky shores in repeating bursts. Tidal surges advanced and retreated through coastal channels carved into bedrock. There were no leaves to rustle, no birds overhead, no insects moving through the air. Near microbial reefs, streams of oxygen bubbles drifted upward through shallow water, creating the faintest trace of biological activity on a world where life flourished everywhere, yet remained invisible from a distance. The shoreline represented the richest expression of Colombia's living world. Beyond it stretched an interior shaped almost entirely by geology, climate, and time. Step away from Colombia's coast, and the planet begins to lose its visible life. The microbial richness of shallow water gives way to exposed stone, broad mineral plains, and dry expanses where the ocean's influence fades with distance. A supercontinent changes climate by its size alone. The farther land stretches from the sea, the harder it becomes for moisture to reach the center. Colombia covered roughly 30% of Earth's current land surface, enough to create an interior desert on a scale difficult to picture. Winds carrying moisture from the surrounding ocean would have dropped much of their water near coastal margins and uplands before reaching the continental center. By the time air masses crossed deeper inland, they were drier, warmer, and less able to support lasting surface water. Large regions may have endured extreme aridity for spans far longer than any human desert has existed. No vegetation softened that world. There were no roots to hold moisture in cracks, no leaves to release water vapor back into the air, no plant cover to shade the ground or slow erosion. Modern inland climates depend partly on biological cycles that recycle water through soils, forests, wetlands, and grasslands. Colombia had none of these systems. Its interior was shaped by rock, sunlight, wind, and the slow movement of minerals through occasional water. The surface faced a harsh sky. With little ozone protection, ultraviolet radiation struck exposed landscapes at levels dangerous to most modern surface organisms. Nothing grew tall enough to cast shade across the ground. Rock absorbed heat during the short day, then released it after sunset as temperatures fell. Without soil or vegetation, there was little buffering between the surface and the atmosphere. The land could swing between punishing heat and cold nights with stark efficiency. Life may still have found narrow refuges. Some modern microbes live inside rocks, only millimeters below the surface, protected from radiation by a thin layer of mineral grains. These organisms, called cryptoendoliths, survive today in places such as Antarctic sandstone and the Atacama Desert. Colombia's interior rock faces may have supported similar communities, living within pores and fractures where faint moisture and chemical traces gathered. To any observer, the surface would appear barren, while life persisted inside the stone. Other habitats existed below ground. In regions where geothermal heat pushed water through fractured crust, subsurface microbial communities could have survived without sunlight. These organisms would have drawn energy from chemical reactions involving minerals, gases, and heated fluids. Colombia's interior was geologically active, with faults, old collision zones, and buried heat sources providing pathways for water and chemistry. Beneath an empty surface, the crust itself may have supported life. Rivers crossed parts of the interior, although they were unlike rivers of later worlds. Rainfall, near margins and highlands, could send water across bare rock, carving channels and carrying sediment. Those rivers transported minerals, sand-sized particles, and dissolved chemicals. They carried no leaf litter, no wood, no pollen, no carcasses, and no soil rich with organic debris. Their beds were channels of moving mineral water. The interior of Colombia may have been among the largest visibly lifeless surface environments in Earth's history. Millions of square kilometers, which is about millions of square miles, stretched under tinted daylight with nothing apparent growing, grazing, nesting, or dying across the open land. Along Colombia's coast, life occupied a space thinner than a fingernail. Entire ecosystems functioned within layers measured in millimeters, yet those layers contained a degree of organization that rivaled anything found in much larger environments. The shallow lagoons, tidal flats, protected embayments, and broad coastal shelves surrounding the supercontinent form the center of Earth's biological activity. Nearly every major ecological process on the planet unfolded here. At first glance, these environments might have appeared as dark films coating submerged rock and sediment. Their true complexity revealed itself only at microscopic scale. A microbial mat was not a uniform sheet of organisms. It was a densely structured community arranged vertically according to chemistry, light availability, and metabolic function. Each layer occupied a narrow zone where conditions matched its needs precisely. The uppermost surface belonged largely to cyanobacteria. Bathed in sunlight, these organisms harvested energy through photosynthesis and released oxygen into the surrounding water. Sticky secretions produced by the cells trapped fine sediment particles drifting through the shallows. As the mat grew upward and accumulated additional material, it reshaped the seafloor grain by grain. This thin, living skin formed the interface between sunlight and the deeper chemistry below. Just beneath that surface existed another photosynthetic community. These bacteria also captured light energy, yet they operated differently. Instead of using water during photosynthesis, they relied on hydrogen sulfide. Their pigments absorbed wavelengths of light that penetrated beyond the cyanobacterial layer above. Oxygen was absent from their metabolism, but solar energy still powered their existence. The same sunlight supported multiple ecosystems stacked within fractions of a centimeter. Below those light-dependent layers, conditions shifted again. Sulfur-reducing bacteria occupied oxygen-free zones where sulfate diffused downward from overlying water. These microbes extracted energy through chemical reactions that produced hydrogen sulfide as waste. That hydrogen sulfide then moved upward and supplied the organisms above. Material flowed continuously between layers, linking each community to its neighbors through a network of chemical exchanges. Deeper still lived methanogenic archaea. In complete darkness, isolated from sunlight and oxygen, they generated methane using carbon dioxide and hydrogen. Their activity formed the lowest layer of a tightly integrated biological system. Waste products from one zone became resources for another. Nutrients circulated through the mat in recurring cycles, creating a self-sustaining economy of chemistry contained within a remarkably small vertical space. The tides governed every aspect of this world. As water retreated, portions of the mat became exposed to air and intense ultraviolet radiation. Cellular activity slowed, protective responses activated, and some organisms entered dormant states. When returning water covered the surface again, metabolic processes resumed. The entire ecosystem expanded and contracted with the rhythm of the shoreline. These communities were not motionless. Cyanobacteria possess the ability to glide across surfaces, adjusting their position according to changing light conditions. Over extended observation, the mat itself would appear to shift and reorganize. Patterns formed, dissolved, and reformed as countless cells responded to environmental changes. What seemed fixed from a distance was actually dynamic and responsive. For hundreds of millions of years before Columbia assembled, ecosystems built on this basic structure had already existed. Across Columbia's coastlines, they continued their patient work, transforming sunlight, minerals, sulfur, methane, and water into one of the most intricate biological systems Earth had yet produced. A strand of red algae drifting through a shallow coastal lagoon would have looked insignificant against the immense backdrop of Columbia's shoreline. Its body was simple, little more than a chain of cells organized into delicate filaments. Yet within organisms like these, one of the most important innovations in biological history was beginning to emerge. The change was not visible in shape, size, or movement. It existed in the way life created the next generation. The fossil, known as Bangiomorpha pubescens, comes from rocks in what is now Arctic Canada and dates to approximately 1.05 billion years ago. By the time these organisms appeared, Columbia had already witnessed countless geological transformations and biological experiments. Cyanobacteria had altered planetary chemistry, complex cells had emerged, new cellular partnerships had reshaped the possibilities of life. Bangiomorpha added another piece to that growing foundation, providing the oldest widely accepted evidence of sexual reproduction in the fossil record. For most of Earth's earlier history, reproduction followed a far simpler path. A cell divided into two descendants. Those descendants divided again. Generation followed generation through replication. Variation still occurred through mutation and the exchange of genetic material between microbes, yet the process remained constrained. Most offspring closely resembled their parents because they were, in essence, copies. Sexual reproduction changed that pattern entirely. Genetic material from two individuals became combined into a new organism, carrying a unique mixture of inherited traits. Every generation introduced fresh combinations that had never existed before. Evolution no longer depended solely on rare genetic accidents appearing one at a time across distant lineages. Existing variations could be shuffled, recombined, and tested continuously. Within populations, this created a dramatic expansion of evolutionary opportunity. Traits that emerge separately could meet within the same lineage. Beneficial adaptations spread through populations more efficiently. Harmful mutations became easier to remove. Diversity increased generation after generation, providing natural selection with a far richer collection of possibilities upon which to act. The significance of this development extended far beyond the algae in which it first appeared. Complex organisms require enormous amounts of genetic information working together in coordinated ways. Different cell types must communicate, specialize, and cooperate. Entire developmental programs must unfold with precision. Such complexity becomes far easier to evolve when genetic variation is abundant and constantly renewed. Across the long span that followed, sexual reproduction became one of evolution's most powerful engines. The biological innovations that would later define plants, fungi, and animals all emerged within lineages capable of generating extensive genetic diversity. Nervous systems, circulatory systems, digestive systems, immune systems, and intricate body plans rested upon foundations built over immense periods of evolutionary experimentation. Sexual reproduction greatly accelerated the supply of raw material needed for that experimentation. Seen from the perspective of Columbia's final chapters, the timing is striking. The supercontinent's world had already produced the nucleus, the mitochondrion, and the chloroplast. Each transformed what cells could accomplish. Near the close of Columbia's existence, sexual reproduction joined that collection of innovations. The essential toolkit required for future biological complexity now existed. The creatures that would eventually use those tools remained far beyond the horizon, yet the machinery itself had already been assembled within the waters surrounding the aging continent. Far below Columbia's surface, beyond the crust and deep into the mantle, heat had been accumulating for hundreds of millions of years. The continent above appeared stable, its coastlines crowded with microbial life, and its interior stretching across immense distances. Yet the foundations of that world were already changing. The same geological forces that had assembled Columbia were beginning to work in reverse. Supercontinents create a problem for the planet beneath them. A landmass of such scale acts like an insulating blanket over the mantle. Heat that would normally escape becomes trapped. As millions of years pass, temperatures rise within the underlying mantle, and that trapped energy seeks a path upward. Columbia had grown broad enough and thick enough for this process to become unavoidable. From great depths, mantle plumes began to rise. These columns of superheated rock moved upward through the mantle, carrying enormous amounts of thermal energy toward the crust. When a plume approached the underside of the continent, the land above responded. The crust arched upward into broad domes. Rocks stretched outward from the center of the uplift. What had once been stable gradually became strained. Fractures appeared across the continent. At first, they were scattered zones of weakness, geological seams opened by persistent tension. As stretching continued, these fractures expanded into rift systems extending across vast regions. The crust became thinner. Magma found pathways upward through newly formed cracks. Columbia was not collapsing in a sudden catastrophe. It was being pulled apart piece by piece through processes operating on timescales far beyond human experience. One of the clearest records of this transformation survives today in northern Canada. There, the Mackenzie Dyke swarm preserves evidence of a major mantle plume event approximately 1.27 billion years ago. Hundreds of ancient magma intrusions radiate outward across the landscape in a giant fan pattern. Some extend for thousands of kilometers, which is about 621 miles. Together, they record a powerful pulse of magma rising through a continent already under stress. As rifting progressed, sections of the crust sank between parallel faults, forming elongated valleys. Water collected within these depressions, creating chains of lakes that stretched across the fractured terrain. Continued thinning allowed seawater to invade some of these low regions. New coastlines appeared where none had existed before. The geography of Colombia became increasingly fragmented as the separation widened. Volcanic activity accompanied much of this process. Lava erupted through weakened crust and spread across the surface in extensive flows. Individual eruptions could continue for long periods, releasing immense volumes of molten rock. Layer upon layer accumulated across portions of the continent, preserving another chapter in the slow dismantling of the supercontinent. Throughout these changes, life responded with remarkable persistence. Fresh shorelines offered new habitat. Shallow marine environments expanded into regions that had previously been dry land. Microbial communities established themselves wherever suitable conditions emerged, spreading across newly flooded margins as naturally as tides crossing a rocky coast. The breakup unfolded over hundreds of millions of years. Colombia did not vanish in a single moment. Fragments drifted apart, oceans opened between them, and the scattered pieces began journeys that would eventually contribute to the assembly of Rodinia. The continent that had dominated Earth was entering its final age, reshaped by forces rising from the planet's interior. The first signs appeared in stone long before ice ever reached the tropics. Coastlines multiplied, continents shifted, and chemical balances that had persisted for immense spans of time began to change. Columbia's fragmentation was transforming the architecture of the planet, yet its most important consequence unfolded through reactions so gradual that no single generation of microbes could have perceived them. As the supercontinent separated into drifting fragments, fresh rock became exposed along expanding continental margins. New shorelines stretched across the globe. Rift valleys flooded. Shallow seas advanced into regions that had once been inland. From a geological perspective, this created an enormous increase in the amount of rock available for weathering. Rainwater plays a subtle role in planetary climate. As it falls through the atmosphere, it absorbs carbon dioxide and forms weak acids. When that water reaches exposed rock, chemical reactions begin. Minerals break down. Elements are released into rivers and eventually carried to the sea. In the process, carbon dioxide is removed from the atmosphere and locked into long-term geological storage. A larger coastline meant more contact between water and rock. More contact meant more weathering. More weathering meant a greater drawdown of atmospheric carbon dioxide. The effect was not immediate. It accumulated across millions of years, gradually weakening one of the greenhouse gases that had helped maintain warmth on Earth, despite a sun that still shone less brightly than it does today. Another transformation was unfolding above the oceans. Throughout Columbia's history, photosynthetic microbes had been releasing oxygen into the environment. For a long time, much of that oxygen was consumed by reactions with dissolved iron and other chemical sinks. As those sinks became increasingly saturated, larger quantities of oxygen remained available in the atmosphere. Oxygen and methane do not coexist peacefully for long. When they meet, methane is broken down through a series of reactions that ultimately produce carbon dioxide and water. Methane had once provided powerful greenhouse warming, molecule for molecule. It trapped far more heat than carbon dioxide. As oxygen concentrations climbed, atmospheric methane steadily declined. Two separate cooling mechanisms were therefore operating at the same time. Weathering was removing carbon dioxide from the atmosphere. Rising oxygen was dismantling methane. The thermal blanket that had insulated the planet for hundreds of millions of years was becoming thinner from multiple directions at once. The process unfolded gradually, yet its consequences accumulated. Global temperatures drifted downward. Ice sheets expanded more readily. Reflective surfaces increased, sending greater amounts of sunlight back into space. Each advance of ice created conditions that encouraged further cooling. Climate systems that had once remained stable moved closer to critical thresholds. Hundreds of millions of years after Columbia's breakup began, Earth entered one of the most extraordinary climatic episodes in its history. During the snowball Earth intervals, beginning approximately 720 million years ago, ice advanced toward equatorial regions and oceans became covered across enormous areas. The world approached a state unlike anything seen before or since. There is a striking irony in this chain of events. The microbial communities that oxygenated the atmosphere were transforming the planet in ways that ultimately undermined the greenhouse conditions under which they had flourished. Through ordinary metabolism, repeated countless trillions of times, they altered atmospheric chemistry, ocean chemistry, and climate itself. Columbia's dissolution had opened the pathway, and biology carried the process forward across the ages. Columbia survives as a puzzle scattered across continents. No map was left behind. No surface from that age remains untouched, and no witness could preserve the shape of its coasts. The supercontinent can only be rebuilt from rock, magnetism, mineral ages, and faint chemical traces locked inside formations older than most mountains now standing on Earth. One of the most powerful tools is paleomagnetism. When molten rock cools, certain magnetic minerals align with Earth's magnetic field and then freeze into place as the rock hardens. That preserved direction acts like a compass needle trapped in stone. By measuring these ancient magnetic signatures, geologists can estimate where a rock once sat relative to Earth's poles. When matching signals appear on continents now separated by oceans, their former positions begin to take shape. This method does not create a picture from imagination. It uses physics. Rocks of the same age can preserve magnetic directions that reveal shared latitude or movement through deep time. When enough measurements align, fragments of crust that now seem unrelated begin to fit together into an older arrangement. Other evidence comes from matching geological provinces. The Eastern Ghats mountain belt of India contains rock sequences, mineral assemblages, structural patterns, and age signatures that closely resemble those found in Enderby Land in Antarctica. These regions are now separated by ocean, yet their rocks suggest they were once joined. Their shared history points back to the configuration of Columbia before later rifting carried them apart. Similar links appear elsewhere. The trans-Hudson origin in North America and the trans-Amazonian origin in South America preserve comparable ages, rock types, and structural architecture. Each records continental collision from the same broad window of time. Today, the South Atlantic Ocean separates them. In Columbia's world, they may have belonged to connected belts formed during the same planetary assembly. Tiny crystals add another layer of evidence. Zircon grains survive erosion, transport, burial, and metamorphism with remarkable durability. Many contain uranium, which decays into lead at a known rate. By measuring that decay, scientists can determine when the crystal formed. Sandstones, carrying zircons from eroded mountains, preserve the age signature of their source regions. Matching zircon populations across continents can reveal mountain belts that once supplied sediment to connected basins. Life also left traces. Organic molecules preserved inside ancient rocks can point toward the organisms that produced them. Some lipid structures are associated with cyanobacteria. Others may indicate early eukaryotic life. Through biomarker geochemistry, scientists can search for chemical fingerprints even where body fossils are absent. These traces help reconstruct the biological setting of Columbia's seas. The idea of Columbia as a supercontinent was formally proposed in 2002 by geologists John Rogers and M. Santosh. Their case drew strength from several lines of evidence converging together, including paleomagnetic data, matching rock provinces, and increasingly precise geochronology. Earlier clues had existed, but the tools needed to combine them convincingly were still developing through the late 20th century. Even now, the reconstruction remains unfinished. Researchers continue to debate the exact positions of Australia, Antarctica, and parts of Asia within Columbia's layout. The broad outline is visible, yet some internal geography remains uncertain. What endures is the deeper achievement, a vanished world rebuilt from stone, one measurement at a time. Far beyond the reach of Columbia's shorelines lies a deeper chapter of Earth's story, one written in fragments so old that much of the original record has been altered, buried, or destroyed. The farther scientists travel into deep time, the fewer clues remain. Even so, scattered pieces of evidence suggest that Columbia may have inherited a pattern already billions of years in motion. One candidate is Kenorland, a proposed supercontinent that may have existed around 2.7 billion years ago. Its reconstruction is built from some of Earth's earliest stable continental cores, including ancestral North America, Baltica, Western Australia, and Karelia. Rocks across these regions share comparable ages and geological histories. Paleomagnetic measurements also indicate positions that may once have placed them together. The evidence remains less complete than the case for Columbia, yet the possibility continues to hold scientific weight because multiple lines of investigation point in the same direction. Further back stands a proposed landmass called Unlike later supercontinents, may have been more modest in scale, functioning as a durable continental nucleus around which future assemblies developed. Geological evidence suggests that parts of southern Africa, Australia, India, and Antarctica preserve crustal fragments that could trace their ancestry to this early structure. If the interpretation is correct, portions of may have survived for billions of years, passing through repeated cycles of collision and separation while retaining a shared geological heritage. Beyond even lies one of the most debated ideas in earth science. Volbara is a hypothetical arrangement based primarily on similarities between the Kapval Craton of southern Africa and the Pilbara Craton of Australia. These regions contain remarkably old rock sequences that display related structural and sedimentary characteristics. Some researchers argue that the similarities are best explained if the two cratons were once joined. Others remain cautious, noting that evidence from such remote intervals is often incomplete. Even so, Volbara remains an intriguing possibility because it would place continental assembly as far back as 3.6 billion years ago. These ancient proposals point toward a larger implication. The process that created Columbia may have begun soon after Earth developed a mobile crust capable of large scale movement. Once plate tectonics became established, continents were no longer fixed features. They drifted, collided, merged, and separated, reshaping the surface again and again across immense spans of time. Geologists describe this repeating pattern as the supercontinent cycle. The cycle unfolds over roughly 400 million to 600 million years. Continents gather into a single landmass, creating a broad, insulating cap over the mantle below. Heat gradually accumulates beneath that crust. Eventually, the pressure becomes sufficient to weaken and fracture the supercontinent. New oceans open, fragments disperse, and the mantle cools. Later, subduction draws those fragments together once more, beginning another phase of assembly. Viewed from that perspective, Columbia was neither a beginning nor an ending. It was one beat within a planetary rhythm already underway long before its formation. Mountains rose and vanished. Oceans opened and disappeared. Continents repeatedly changed partners across spans of time almost beyond comprehension. That rhythm has never stopped. The same tectonic engine continues operating beneath the modern world, carrying continents across Earth's surface and preparing conditions for future configurations still far beyond the horizon of human history. A ridge of volcanic rock runs down the middle of the Atlantic Ocean, far below the waves. Along that boundary, molten material rises from Earth's interior and hardens into new seafloor. The process is occurring now, at this moment, just as it did during ages long before humans existed. The forces that assembled Columbia belong to deep time, yet they have never stopped operating. The Atlantic Ocean continues to widen at roughly 2.5 centimeters per year, which is about 1 inch per year, a pace comparable to the growth of a human fingernail. North America and South America drift gradually westward. Europe and Africa move away in the opposite direction. The separation is almost impossible to notice within a human lifetime, yet over millions of years, the cumulative movement reshapes the globe. Across the planet, a different story is unfolding. The Pacific Ocean is shrinking. Around much of its perimeter, dense oceanic crust descends into the mantle through subduction zones. More crust is being consumed than created. Each year, the basin loses a little more ground, continuing a trend that has persisted for tens of millions of years. Project those motions far enough into the future, and familiar maps begin to dissolve. Many geological models suggest that within roughly 200 million to 250 million years, the Pacific may close completely. Continents that now face one another across the world's largest ocean could eventually collide. Mountain ranges would rise where seafloor once existed. New continental interiors would emerge where open water now stretches to the horizon. Scientists have proposed more than one destination for that future journey. One model, known as Amasia, envisions continents gathering around the northern polar region as the Arctic Ocean closes and land masses rotate northward. Another scenario, called Pangaea Proxima, suggests that after the Pacific disappears, tectonic changes could eventually reverse the Atlantic's expansion, drawing continents back together near equatorial latitudes. The details remain uncertain, yet the broader pattern is familiar. When a new supercontinent forms, it will inherit many of the same physical challenges that shaped Columbia. A continental interior, far from moisture sources, would likely become intensely arid. Ocean circulation would change. Atmospheric systems would reorganize. Deep beneath the crust, Mantle heat would accumulate under the insulating blanket of merged continents. That accumulated heat matters. Supercontinents are temporary arrangements because the planet beneath them remains active. Rising mantle material gradually weakens the crust from below. Fractures appear. Rift valleys develop. New oceans eventually open. The cycle of assembly and dispersal begins again. Earth's interior is cooling across geological time, yet the process is proceeding with extraordinary slowness. Current evidence suggests that plate tectonics may continue for at least another billion years, perhaps considerably longer. Continents will keep moving. Oceans will keep opening and closing. Future worlds will inherit landscapes impossible to predict in detail. Columbia stands as the earliest confirmed chapter of this continuing rhythm. Its rocks preserve proof that the supercontinent cycle was already operating at least 1.8 billion years ago. The continents beneath our feet remain participants in that same story, drifting through a sequence that will continue long after every city, language, and species alive today has passed into the geological record. Imagine lowering a microscope into one of Colombia's coastal waters and watching life unfold across a landscape measured in fractions of a millimeter. Activity would be everywhere. Cells would divide, chemical gradients would shift, microbial communities would expand across sunlit surfaces. Entire ecosystems would operate with astonishing sophistication. Yet within all that activity, there would be no experience of danger. The living world of Columbia contained organisms capable of harvesting sunlight, processing sulfur compounds, producing methane, consuming dissolved nutrients, and transforming the chemistry of oceans and atmosphere. Many belonged to lineages already refined by hundreds of millions of years of evolution. Complexity existed in abundance. What did not exist was perception. A modern reef is filled with signals. Fish detect movement. Crustaceans react to vibration. Predators search for prey while prey search for shelter. Countless nervous systems remain engaged in an ongoing negotiation with risk. Columbia belonged to a different biological reality. No organism possessed a brain. No organism possessed a nerve. No organism formed an internal representation of its surroundings. Environmental change produced biochemical responses, yet nothing interpreted those responses as events. Competition still shaped evolution. Colonies occupying shallow water absorbed light that neighboring colonies could no longer reach. Microbes consumed nutrients before competitors could access them. Populations expanded into favorable conditions and declined when resources became scarce. Selection remained active, sculpting genomes generation after generation. The process resembled chemistry unfolding across planetary scales, not conflict as later life would understand it. Viruses moved through these communities as well. They entered cells, hijacked molecular machinery, and often destroyed their hosts. Certain bacteria consumed other microorganisms. Genetic material passed between unrelated lineages. Entire populations rose and fell. Through all of it, there was no alarm, no anticipation, and no attempt to escape. Biological consequences existed without biological awareness. This distinction matters because many features associated with life today emerged only after organisms began responding behaviorally to one another. Speed becomes valuable when something can chase you. Armor becomes useful when something can bite. Camouflage matters when being detected carries consequences. Venom, warning colors, mimicry, ambush tactics, pursuit strategies, and coordinated defense all belong to a world shaped by perception and reaction. Colombia's ecosystems existed before that transformation. Near the later stages of this era, some eukaryotic organisms likely began grazing upon smaller cells. Those interactions represented an early step toward behavior driven ecology. Even then, the process remained simple compared with the predator and prey relationships that would emerge much later. The oceans still lacked the elaborate evolutionary contests that would eventually dominate animal life. For hundreds of millions of years, the planet functioned under these conditions. Life spread across coastlines, altered atmospheric chemistry, reshaped marine environments, and persisted through continental change. It achieved extraordinary durability while remaining untouched by fear, pain, pursuit, or hunger in the animal sense. Metabolism governed every interaction. Molecules flowed through cells. Energy passed from one process to another. Time accumulated across generations beyond counting. Viewed from that perspective, Colombia's biosphere invites a different measure of success. Its inhabitants never built forests, developed intelligence, or left footprints in stone. Even so, they endured, expanded, and transformed an entire planet, achieving a longevity and stability that few later forms of life would ever match. Success is often imagined as complexity: forests spreading across continents, animals crossing oceans, civilizations rising from stone. Viewed through the lens of deep time, another story emerges, one written by organisms so small that billions can fit within a single handful of seawater. During Columbia's era, every living thing on Earth belonged to the microbial world. No trees cast shadows, no animals crossed shorelines, no roots fractured rock. Every gram of living biomass on the planet consisted of bacteria, archaea, and their microbial relatives. The biosphere was entirely theirs. Measured by persistence, their achievement is difficult to overstate. Evidence places microbial life on Earth at least 3.5 billion years ago. Complex multicellular organisms arrived far later. Animals, plants, and fungi occupy only the most recent fraction of the planet's biological history. For most of Earth's existence, microbes were not preparing the stage for larger forms. They were the main story. Their dominance continues even now. Modern estimates suggest that bacteria and archaea together account for a substantial portion of Earth's total biomass by carbon. In Columbia's time, that share reached 100%. Every coastline, every shallow lagoon, every fracture carrying mineral-rich water through rock belonged to microbial ecosystems. Life and microbial life were effectively the same thing. Their record of survival is equally striking. Earth has endured repeated planetary crises. Asteroid impacts altered climates. Massive volcanic eruptions transformed atmospheric chemistry. Oceans lost oxygen. Ice advanced across enormous portions of the globe. The great biological catastrophes remembered for erasing entire groups of animals passed through microbial communities with comparatively limited effect. Species changed. Populations shifted. The foundation remained intact. This perspective challenges a familiar assumption. Evolution is often described as a ladder leading upward from simple forms toward increasingly elaborate organisms. The rocks tell a different story. Evolution has no preferred destination. Natural selection rewards whatever persists. By that measure, microbial life occupies a position of extraordinary success. A cyanobacterium drifting through Colombia's coastal waters may appear simple when compared with a mammal or a bird. Appearances conceal an astonishing level of molecular sophistication. Within that tiny cell operated intricate protein assemblies capable of capturing sunlight and directing its energy through precisely coordinated chemical pathways. One of those systems, known as photosystem 2, could separate water into its constituent elements, using light itself as the driving force. Human technology still struggles to imitate that achievement efficiently under ordinary conditions. The cyanobacteria of Colombia performed it continuously, generation after generation, while simultaneously reshaping oceans and atmosphere on a planetary scale. Seen from enough distance, complex life begins to resemble a recent branch growing from a much older trunk. Every animal that has ever hunted, flown, swum, burrowed, or walked exists atop a foundation established by microbial innovators long before the first muscle contracted or the first eye formed. Columbia captures a moment when that foundation stood alone. The planet belonged entirely to microscopic organisms whose influence extended far beyond their size. They transformed chemistry, altered climate, engineered habitats, and endured across spans of time that dwarf the history of every animal lineage combined. In the language of deep time, their record remains unmatched. Place a hand against your chest and consider the activity unfolding beyond perception. Cells are dividing, proteins are folding into precise shapes. Energy is moving from molecule to molecule. Blood is carrying oxygen through branching vessels. Every one of these processes depends upon biological systems whose origins reach back to the world of Columbia. The connection is neither symbolic nor poetic. It is physical. The inheritance is written into the structure of every human cell. Within most cells of the body reside mitochondria, tiny organelles responsible for supplying usable energy. Their ancestry traces back to free-living alpha proteobacteria that entered into partnership with larger cells during Columbia's era. That partnership became permanent. Across roughly 1.5 to 2 billion years, descendants of those ancient bacteria continued reproducing inside their hosts. They remain there today. Evidence of that history survives in their biology. Mitochondria possess their own deoxyribonucleic acid, separate from the deoxyribonucleic acid contained within the nucleus. That genetic material forms a circular structure, a characteristic shared with bacteria. Mitochondria multiply through division, producing copies of themselves in a manner inherited from microbial ancestors. Every cell carrying mitochondria preserves traces of an event that occurred long before animals, plants, or fungi appeared. The line of inheritance extends even further. Mitochondrial deoxyribonucleic acid passes through the maternal line, generation after generation. Mothers transmit mitochondria to their children. The chain stretches backward through human history, through primates, mammals, and earlier vertebrates, eventually reaching the first eukaryotic cells that carried these microbial partners. No break exists anywhere along that sequence. Energy production itself reveals the same continuity. The biochemical pathways operating inside human cells belong to a framework established in distant oceans. The citric acid cycle, oxidative phosphorylation, and the electron transport chain form part of a deeply conserved metabolic architecture. Countless modifications have accumulated through evolutionary time, yet the underlying logic remains recognizable. Human cells still rely upon molecular machinery, whose foundations were laid in microbial ecosystems of immense antiquity. Each breath reflects another inheritance. Oxygen fills the atmosphere today because generations of cyanobacteria gradually transformed the planet's chemistry. During Columbia's era, those microorganisms continued releasing oxygen into the oceans and sky, contributing to conditions that later forms of life would depend upon. Every inhalation participates in a cycle they helped establish. Even the iron carried within hemoglobin belongs to a story that reaches into that distant world. Long before blood existed, iron and oxygen were already interacting across marine environments, producing chemical relationships that would later become central to animal physiology. The same element moving through human circulation today once participated in planetary reactions unfolding across Columbia's seas. The inheritance extends into the organization of the genome itself, regulatory systems controlling gene expression, molecular mechanisms responsible for editing genetic instructions, and proteins that package deoxyribonucleic acid into manageable structures emerged within early eukaryotic lineages. Those innovations persisted, expanded, and diversified, eventually supporting organisms of extraordinary complexity. For that reason, Columbia occupies more than a chapter in Earth's history. Its legacy remains active in every heartbeat, every breath, and every cell. The continent vanished long ago. The biology it helped shape never left. A number can be spoken in a single breath and still remain almost impossible to comprehend. 1.8 billion years belongs to that category. The words arrive easily, the reality behind them does not. Human intuition evolved to understand seasons, lifetimes, and generations. Deep geological time exists far beyond those scales, requiring a different kind of imagination. One way to approach it is through compression. Imagine Earth's entire history, from its formation 4.54 billion years ago to the present day, condensed into a single calendar year beginning on the first day of January. Continents assemble and disperse. Oceans appear and vanish. Atmospheres change. Life emerges and transforms. Within that calendar, Columbia would have assembled near the end of February. For months afterward, the planet would remain a microbial world. Spring, summer, and much of autumn would pass without forests, animals, flowers, insects, or fish. The first animals would not arrive until the middle of November. Dinosaurs would appear on the 13th day of December. Their disappearance would occur only a few days before the year ended. Human beings occupy an even narrower interval. Anatomically modern humans emerge during the final minutes of the 31st day of December. Agriculture arrives seconds before midnight. Cities, kingdoms, writing systems, and recorded history crowd together near the very end. Every empire, every voyage, every war, every library, every scientific theory, every machine, and every satellite fits within roughly the final 10 seconds before the clock strikes 12. Against that backdrop, Columbia assumes a different scale. Estimates suggest the supercontinent persisted for roughly 300 million to 500 million years. On the compressed calendar, that corresponds to several weeks. Its lifespan exceeds the entire history of animals living on land. What appears distant from a human perspective becomes enormous even when measured against evolutionary milestones. Certain living structures help bridge that gap. Along the shores of Shark Bay in Western Australia stand stromatolites built by microbial communities. Visitors can walk near them today. Sunlight falls across their layered surfaces. Waves move around them. Their architecture and biological function closely resemble formations that existed during Columbia's time. The interval separating them spans nearly 2 billion years, yet the underlying strategy endured. That persistence carries its own lesson. Entire branches of animal life appeared and vanished, while these microbial builders continued occupying ecological niches that remained available. Continents shifted positions. Mountain ranges rose and eroded away. Ice ages advanced and retreated. Through all of it, certain microbial lineages maintained ways of living that required little alteration. Deep time, therefore, changes perspective in more than one direction. It reveals how recent human civilization truly is, while also emphasizing the durability of some biological solutions. Complexity attracts attention because it resembles us. Longevity tells a different story. A lineage surviving for billions of years has achieved something extraordinary, even if it never developed eyes, limbs, or brains. Seen from that vantage point, Colombia becomes more than a lost supercontinent. It becomes a marker within a span so immense that human history occupies only the thinnest edge of the frame, a brief flicker at the end of a year that began long before memory and will continue long after it. When people imagine life beyond Earth, the mind often drifts toward creatures, landscapes, or civilizations. Colombia offers a different perspective. For most of our planet's biological history, life did not announce itself through bodies large enough to see. It worked through chemistry, persistence, and numbers beyond counting. Measured by longevity, influence, and planetary reach, microbial life remains the dominant form of life that Earth has ever known. Animals, plants, and fungi occupy only the most recent chapter. For more than 3 billion years, every living thing on Earth belonged to the microbial world. Oceans, sediments, coastlines, and the deep crust were inhabited exclusively by organisms whose existence would have escaped human notice. That reality matters when considering the possibility of life elsewhere. If Earth serves as our only confirmed example, then the strongest statistical lesson from its history is straightforward. Life spends most of its existence in microscopic form. Columbia's world was not a transitional curiosity waiting for something larger to arrive. It was a stable and successful biosphere that endured for immense spans of time. Its inhabitants harvested sunlight, consumed minerals, exchanged electrons, recycled nutrients, and reshaped atmospheric chemistry. They transformed the planet without eyes, limbs, or nervous systems. A distant world supporting comparable organisms could appear lifeless from orbit while hosting an active biological system operating across entire oceans. Several places within our own solar system now appear capable of providing conditions similar to those that sustained microbial ecosystems on early Earth. One of the most compelling is Europa, a moon orbiting Jupiter. Beneath its frozen surface lies a global ocean. Gravitational forces generated by Jupiter continuously flex the moon's interior, producing heat that helps keep water in liquid form. Many researchers suspect that interactions between seawater and rocky seafloor materials generate chemical gradients capable of supporting metabolism. Another candidate circles Saturn. Enceladus releases towering plumes of water vapor from fractures in its icy crust. Measurements collected by the spacecraft named Cassini revealed organic compounds, silica particles, and molecular hydrogen within those plumes. Together, these findings point toward active hydrothermal systems below the surface. Such environments resemble settings where microbial communities flourish on Earth today. Mars presents a different possibility. Geological evidence indicates that rivers, lakes, and perhaps even large bodies of standing water existed during its distant past. As conditions became colder and drier, any potential biosphere would have faced immense pressure. Yet microbial organisms on Earth routinely survive within rock pores, underground aquifers, and chemically active fractures. If life emerged on early Mars, some researchers consider it plausible that descendants could persist below the surface. Columbia encourages a practical approach to this search. The most valuable clues may not be structures, artifacts, or technological signals. They may be subtle chemical fingerprints preserved in rock, dissolved in water, or trapped within ice. Scientists studying Earth's distant past already rely on such evidence. Ancient molecules, isotopic ratios, mineral alterations, and metabolic byproducts reveal the presence of organisms long after their bodies have vanished. Viewed through that lens, the search for life elsewhere becomes less a hunt for something familiar and more an effort to recognize the signatures of metabolism. Columbia's world demonstrates that a planet can be profoundly alive while remaining almost entirely invisible from a distance. For hundreds of millions of years, waves crossed Columbia's shores, continents shifted position, mountains rose and wore away, and the atmosphere changed composition. Throughout all of it, there was no witness. Life covered coastlines, filled sediments, occupied the water column, and transformed planetary chemistry. Yet, nothing living possessed the capacity to know any of it was happening. No organism watched the sun rise above Columbia's horizon. No organism recognized the passage of seasons. No organism observed oxygen accumulating in the atmosphere molecule by molecule. There was metabolism, growth, reproduction, competition, and adaptation, but there was no awareness attached to those processes. The world existed in extraordinary detail, though no living thing could form a thought about its existence. That absence makes the present moment unusual in a way that is easy to overlook. Columbia vanished more than 1 billion years ago. Its mountains have been eroded, buried, altered by heat, and scattered across continents. Its oceans disappeared long before the first animal walked on land. Nothing remains in its original form. Yet, despite that distance, its outline has become visible again. Fragments of metamorphic rock preserve traces of continental collisions. Ancient magnetic minerals retain records of vanished latitudes. Iron-rich layers reveal shifts in ocean chemistry. Organic molecules trapped within stone preserve signatures of organisms whose bodies dissolved long ago. Piece by piece, evidence separated by continents and immense spans of time can be assembled into a coherent story. This achievement belongs to a very recent development in Earth's history, somewhere within the long chain of descent that began with microscopic cells. Nervous systems emerged. Those nervous systems increased in complexity across millions of generations. Memory, learning, planning, language, and abstraction gradually appeared. Eventually, a species arose capable of asking where it came from and then devising methods to investigate the answer. Viewed from the perspective of deep time, that transition is extraordinary. For most of life's existence, no organism could reconstruct the past. No organism could calculate the age of a rock, measure radioactive decay, compare fossils across continents, or infer the existence of vanished worlds. Such abilities occupy only the latest fraction of the biological timeline. The lineage that acquired them did not arrive from outside the story. It emerged from the same evolutionary foundation established during Columbia's age. The early eukaryotic innovations discussed throughout this journey eventually gave rise to organisms capable of perception. Perception led to increasingly sophisticated cognition. Cognition produced curiosity, and curiosity produced science. That chain of events allows Earth, in a sense, to become aware of portions of its own history. Through observation, measurement, and reasoning, living matter examines the conditions from which it emerged. The rocks themselves do not speak, yet information stored within them can be translated into narrative. A final contrast remains. The cyanobacteria that altered atmospheric chemistry during Columbia's time followed the constraints of metabolism. Their actions changed the planet, though they possessed no understanding of the consequences. Human beings occupy a different position. We can recognize the relationship between life and environment. We can study it, model it, and anticipate outcomes before they occur. The ability to understand carries responsibilities that no earlier organism possessed. It is a capability born from the same deep history we have been exploring, a history that transformed a microbial world into one capable of reflecting upon itself. Few names in geology have aged as awkwardly as the phrase boring billion. It sounds like a judgment passed on an era of stagnation, a stretch of planetary history where little happened and life drifted through a long interval of repetition. For many years, that interpretation seemed reasonable. Rocks from roughly 1.8 billion to zero point 0.8 billion years ago appeared to show the same broad ecological patterns, persisting with little visible change. The impression came largely from fossils. Layer after layer revealed familiar microbial forms. The atmosphere remained relatively stable. No forests emerged. No animals appeared. No dramatic expansion of body plans transformed the biosphere, compared with the spectacular revolutions that came before and after. This interval looked subdued. Generations of researchers inherited the label, and it remained attached to the period even as new evidence began to accumulate. Part of the problem lies in preservation. The dominant organisms of the time were microscopic, soft bodied, and poorly suited for fossilization. Their remains were often destroyed before they could enter the geological record. Many surviving rocks from this interval later experienced heat, pressure, and chemical alteration. Information that once existed within them was erased, distorted, or scattered. A sparse fossil archive can create the illusion of biological stillness, even when change is occurring beyond our ability to observe it directly. Different tools began revealing a more complicated story. Molecular clock studies compare genetic differences among living organisms and estimate when their evolutionary lineages diverged. Those calculations consistently point towards substantial biological innovation during this era. Numerous eukaryotic groups appear to have separated from common ancestors throughout this period, establishing branches that would later flourish across the planet. Physical discoveries have gradually supported that picture. Microfossils from the Grassy Bay Formation in Arctic Canada preserve cellular features dated to approximately 1.9 billion years ago. Carbon-rich compressions from the Ruyang Group in China, dating to about 1.7 billion years ago, contain evidence of internal cellular organization associated with eukaryotic complexity. These remains are fragmentary, yet they hint at communities more sophisticated than earlier interpretations suggested. The rocks themselves carry additional clues. Ratios of different isotopes preserved in sediments reveal fluctuations in ocean chemistry and atmospheric conditions. Those chemical records indicate recurring oxygenation events, environmental instability, and ecological shifts that left little trace in conventional body fossils. Entire chapters of biological activity may have unfolded without producing structures durable enough to survive into the present. Viewed through this broader lens, the interval begins to resemble a workshop rather than a pause. Eukaryotic cells were refining new ways of living. Sexual reproduction was becoming more widespread. Experiments in multicellularity appear to have emerged repeatedly. Lineages were diversifying, adapting, and interacting across oceans whose details remain only partially visible through the surviving evidence. The apparent stillness belonged largely to the limitations of observation. Earth was not resting. Evolution was continuing its work at microscopic scales, shaping possibilities that would become visible much later. What once appeared uneventful increasingly looks like a prolonged phase of preparation, a span of time during which biological complexity expanded beyond the resolution of the fossil record available to us. A span of approximately 800 million years separated Columbia's assembly from one of the most transformative intervals in the history of life. Across that immense gulf of time, oceans changed, continents shifted, climates fluctuated, and lineages diverged. Then, around 540 million years ago, the fossil record brightened with a burst of biological complexity unlike anything that had come before. Within a geological interval estimated at roughly 20 to 25 million years, a brief moment by the standards of deep time, an extraordinary variety of animal forms appeared. Organisms emerged bearing structures that would define much of later evolution. Eyes capable of detecting light and movement entered the story. Limbs and appendages began interacting with the environment in new ways. Shells and exoskeletons offered protection and support. Digestive tracts processed food through specialized internal pathways. Muscles powered active movement. Nervous systems coordinated behavior. Circulatory systems distributed resources. Immune systems distinguished self from intruder. Features that seem familiar today arrived in a comparatively compressed interval. This event is known as the Cambrian explosion. Scientists continue debating the precise combination of triggers behind it. Environmental change, ecological interaction, oxygen availability, developmental innovation, and genetic opportunity all appear to have played important roles. Discussion continues because the event was complex, with many causes interacting across millions of years. What remains far less controversial is the foundation that made such an expansion possible in the first place. The essential groundwork had been laid long before the first Cambrian animals appeared. During Columbia's world, eukaryotic cells emerged and diversified. These cells possessed an internal architecture capable of regulating large numbers of genes with precision. Compartments separated biological tasks. Information could be managed, copied, and expressed through increasingly sophisticated systems. The blueprint required for complex bodies began taking shape within organisms invisible to the naked eye. Energy formed another crucial part of the story. The incorporation of the bacterial ancestors of mitochondria provided cells with access to far greater energetic capacity. Active tissues demand substantial power. Muscle contraction, rapid signaling between nerve cells, immune responses, and coordinated development all require large and reliable energy supplies. The partnership established during Columbia's era expanded what life could afford to build. Photosynthetic innovations from that distant world also mattered profoundly. Lineages carrying chloroplasts transformed sunlight into biological productivity while contributing to the long process of oxygen enrichment. Over immense stretches of time, oxygen became increasingly available to support metabolically demanding organisms. Active animals required conditions that earlier worlds could not easily provide. Sexual reproduction added another layer. By reshuffling genetic information generation after generation, it increased variation within populations. New combinations could be tested, refined, and preserved through natural selection. When ecological opportunities expanded, evolution possessed a powerful mechanism for exploring them rapidly. Seen from this perspective, the Cambrian explosion resembles the visible flowering of systems assembled much earlier. The genetic framework, cellular organization, energetic machinery, oxygen-rich environment, and evolutionary flexibility were already in place. Columbia's oceans hosted many of the innovations that later enabled complex animals to emerge. The dramatic appearance of Cambrian life did not arise from an empty stage. It grew from foundations constructed across hundreds of millions of years, foundations established by organisms whose influence extended far beyond their own age. There is a tendency to imagine deep history as a series of closed chapters. One world ends, another begins, and the boundary between them feels complete. Columbia is often placed into that mental category, a vanished planet inhabited by unfamiliar organisms under skies that belong to another age. The farther back we travel, the easier it becomes to treat the past as disconnected from the present. Yet Columbia resists that interpretation. A lost world is something severed. Its inhabitants disappear, its chemistry fades, its influence survives only as a memory preserved in stone. Columbia followed a different path. The continents that formed it eventually separated. Mountain ranges eroded. Oceans changed shape. Biological communities transformed. Through all of that change, the underlying continuity remained intact. Consider the activity occurring inside a single human cell. Mitochondria continue generating energy through a partnership first established during Columbia's era. They divide using processes inherited from bacterial ancestors that entered another cell and stayed across approximately 1.5 to 2 billion years. That relationship has persisted without interruption. Every heartbeat depends upon it. Every thought depends upon it. Every movement depends upon descendants of a biological merger that occurred in oceans unimaginably distant in time. The atmosphere tells a similar story. Oxygen did not appear once and then remain unchanged. Generation after generation of photosynthetic organisms continued adding it to the planet's surface environment. Cyanobacteria living today perform the same fundamental task carried out by their distant predecessors. The oxygen entering human lungs belongs to a cycle maintained across nearly 2 billion years of continuous biological activity. Even familiar components of the body carry traces of that inheritance. Iron atoms move through hemoglobin exactly as chemistry requires, binding and releasing oxygen according to principles established long before animals existed. The relationship between oxygen and iron that shaped Columbia's seas still operates within blood vessels. Matter cycles, structures evolve, yet the underlying chemistry remains recognizable across immense stretches of time. In Western Australia, stromatolites continue growing in shallow coastal waters. Their layered forms resemble structures that bordered ancient shorelines during Columbia's existence. They are not fossils reenacting a vanished way of life. They are living participants in a lineage that never disappeared. The passage of eras altered the world around them, while the fundamental strategy endured. Viewed this way, continuity becomes more striking than change. Extinctions occurred, continents assembled and dispersed, entire ecosystems appeared and vanished. Through every transition, life maintained an unbroken chain linking one generation to the next. No gap separates the present biosphere from that distant world. Each organism emerged from predecessors, which emerged from earlier predecessors, extending backward without interruption. The thread connecting Columbia to the present was never cut. It stretched through shifting climates, through drifting continents, through the rise of complex organisms and the arrival of minds capable of reflecting on their origins. What appears distant in time remains physically present in living systems. The world of Columbia did not disappear into history. It transformed, adapted, diversified, and continued forward. The same planetary experiment persisted through every age that followed. The forms changed, the connection did not. A shoreline can exist for 1 billion years without ever being seen. Waves can rise and fall. Minerals can circulate through water. Cells can divide, adapt, exchange genes, and reshape a planet's chemistry. Entire chapters of planetary history can unfold without a single living thing, possessing the ability to recognize that anything is happening at all. For the immense span of Columbia's existence, that was the condition of the world. The orange atmosphere received the light of the sun each day. Iron moved through coastal waters and settled into layers that would endure across ages. Broad microbial communities spread across shallow seas, releasing oxygen into an environment that was being transformed molecule by molecule. Tides advanced and retreated under the influence of a nearer moon. Ultraviolet radiation poured across exposed surfaces. Within countless microscopic cells, biological innovations accumulated. None of these events entered a memory. None became a story. No mind existed to assemble experience from the flow of chemistry. This absence of observation was not unusual. It was the normal state of the universe. Stars ignite within clouds of gas and spend millions or billions of years converting matter into light. Galaxies collide and merge, oceans appear on young planets, continents gather and separate. Throughout most of cosmic history, these processes unfold without record or audience. Nature proceeds according to physical law, independent of recognition. Against that backdrop, the appearance of awareness becomes an unusual development. The ability to perceive, remember, compare, and understand arrived very late. Long after Columbia's seas had changed, long after many of its landscapes had vanished, long after the first eukaryotic cells began their journey through evolutionary time. What makes this remarkable is the source of that awareness. The capacity to reconstruct Columbia did not descend from somewhere outside its history. It emerged from the same chain of life that Columbia sustained. The cells that eventually formed nervous systems carried inheritances assembled during that distant era. Those nervous systems eventually supported language, memory, mathematics, geology, and science. Step by step, generation by generation, the biological experiment expanded its ability to examine itself. A person studying an ancient rock formation today is therefore connected to the world being studied in a direct biological sense. The observer and the observed belong to the same continuous lineage. The oxygen supporting thought traces back through the same planetary processes being investigated. The cellular machinery enabling reflection descends from innovations established in those remote oceans. Seen from that perspective, awareness of deep time becomes one of deep time's own products. Columbia did not merely alter oceans, atmospheres, and ecosystems. Through a chain of events stretching across nearly 2 billion years, it contributed to the emergence of organisms capable of asking where they came from and then developing the tools required to answer. Iron formations, oxygen accumulation, and cellular complexity remain among the great achievements of that world. Yet another outcome stands alongside them. From a planet once filled entirely with life that could not witness itself, emerged a lineage able to look backward across immense spans of time and recognize the story written there. The only witness Columbia ever had was created by Columbia itself. Some foundations become so familiar that they disappear from notice. A person can walk across a landscape, breathe the air, touch stone, cross oceans, and live an entire life without realizing how much of the present rests upon events that unfolded almost 2 billion years ago. The world appears complete in itself, yet every living moment is supported by structures assembled long before any animal existed. Columbia's mountains have long since lost their original form. Their peaks were worn down, their sediments carried elsewhere. their roots buried beneath younger layers of rock. Coastlines vanished as continents shifted position across the globe. The atmosphere changed color. The chemistry of the oceans changed with it. One world yielded to another, and then another after that. Even so, traces remain everywhere. Across northern regions of Canada, exposed expanses of the Canadian Shield still preserve fragments of crust that formed during those distant ages. Glaciers later swept across them, stripping away younger coverings and revealing rock that began its journey when life was still confined to microscopic forms. The stone visible today is part of a story that extends beyond imagination, carrying memories written in minerals rather than words. Far away in Western Australia, stromatolites continue growing in shallow coastal water, layer by layer. Microbial communities build structures closely resembling those produced by their predecessors across immense spans of time. Nearby rocks preserve records reaching back more than 3 billion years. The continuity is not symbolic, it is physical, measurable, and ongoing. Columbia also travels in forms less obvious. Iron extracted from ancient deposits becomes bridges, ships, railways, and buildings. Many of those deposits owe their existence to biological and chemical interactions established during that remote chapter of Earth history. Materials supporting modern civilization often began their journey in environments that no longer exist. The atmosphere carries another inheritance. Every breath depends upon oxygen, maintained through a biological process whose roots stretch deep into the past. Generation after generation, countless organisms have continued that work without interruption. The air surrounding every city, forest, desert, and coastline remains linked to metabolic pathways that transformed the planet long ago. Within the human body, the connection becomes even more direct. Tiny energy producing structures inside cells continue reproducing as they have for immense spans of evolutionary time. Their ancestry reaches back through the earliest eukaryotic organisms. The machinery sustaining thought, memory, movement, and awareness depends upon relationships forged in seas that disappeared before complex life emerged. Seen this way, Colombia is neither a destination in the past nor a vanished world separated from the present. It exists as a deeper layer of reality, a geological and biological foundation supporting everything that followed. The surface has changed many times, the underlying thread endured. Tonight, after nearly 1,800,000,000 years, one branch of that continuous lineage can look outward and inward at the same time. The rocks remain, the chemistry remains, the living machinery remains, and from within that continuity comes the rare ability to know that it happened at all. That knowing is part of the same story.